Buenos días. Antes de iniciar, les recordamos que las puertas en ambos lados del auditorio son salidas de emergencia, por lo que hay que dejar las pasillas libres. Si suena una alarma, diríjanse a la salida más cercana. Hay un punto de reunión en el estacionamiento del Jardín Botánico. Bienvenidos al seminario del Instituto de Biología. Hoy me da mucho gusto presentar al Dr. Michael Peruganan. El Dr. Peruganan obtuvo su licenciatura en química de la Universidad de Filipinas en Diliman y realizó sus posgrados en la Universidad de Columbia y la Universidad de Georgia. Su línea de investigación se centra en la genómica evolutiva de las plantas, especialmente en la genómica evolutiva de los cultivos. Es el Silver Professor de Biología en el Centro de Genómica y Biología de la Universidad de Nueva York y también es profesor afiliado del Instituto de Estudio del Mundo Antiguo del campus de la NYU en Abu Dhabi. Actualmente se desempeña como director académico de 19 Washington Square North y anteriormente fue decano de ciencias de la NYU. Ha sido becario Guggenheim y Kavli, investigador joven de Sloan, y fue elegido miembro de la American Association for the Advancement of Science, miembro correspondiente del National Academy of Science and Technology de Philippines, y el año pasado fue elegido miembro del US National Academy of Sciences. Esta primavera de 2025 será profesor visitante en el All Souls College de la Universidad de Oxford. El título de su ponencia es The Geographical Spread of Crops. Bienvenidos.
Gracias. Can you hear me? Soy filipino, pero mi español es muy mal, so I will be speaking in English, unfortunately. But it's always a pleasure to be here at UNAM. I first came to UNAM in 1999, I think. And I think I was here last November in the Frontera Symposium. So I met several of you there, and I think I told a story where I actually, in the late 90s, I almost moved to UNAM. I was really seriously thinking about moving from North Carolina to here. That didn't happen, but with Trump as the president, I may seek asylum here in Mexico, me and my wife. So when I was here in the Frontera Symposium last November, I talked about, in general, about domestication of crops and the evolutionary process of crop evolution. Today, I'm going to talk more about some work that we've been doing over the last 10 years on how especially one crop that you'll see later on, rice, has evolved and has spread geographically after its evolution. And I think by learning more about this, we can learn more about some of the evolutionary processes associated with species range expansions. So just again, to give you a little bit of an introduction, one of the reasons that we work on domestication and domesticated crops is motivated by Charles Darwin. And as I mentioned the last time, and as you know, Darwin was very much interested in domesticated species. He used domesticated species as a way to understand both the nature of variation and the nature of selection. And in the introduction to Origin of Species, which he wrote, he said, there's no reason why the principles which have acted so efficiently under domestication should not have acted under nature. And so what we're trying to do in my laboratory, one of the things we're trying to do is to try to understand the evolutionary process, the same way that Darwin did by looking at domesticated species as well. And just to remind you, there's actually a lot of debate on how we define what domestication is. To me, domestication really is a co-evolutionary process that arises from a unique mutualism. It's a mutualism involving two populations or two species, a domesticator and a domesticate. The domesticator actively manages the fitness of the domesticate population, in this case, a crop, for example, because that domesticate gives resources or services back to the domesticator. And humans are the ones that we are most familiar with as domesticators, but there are also a number of insect species that domesticate fungal species. So it's not something that's associated solely with human species. But in humans, we've domesticated crops, we've domesticated livestock as well. And so this is the definition, just to give you the full definition. This is from this paper I wrote in Trends in Ecology and Evolution 2 years ago. So domestication is a co-evolutionary process that arises from a mutualism in which one species constructs an environment where it actively manages both the survival and reproduction of another species in order to provide the former with resources and or services. So it's a unique mutualism because of the fact that one species is trying to actively manage the fitness of another because it's getting something out of it directly from that active management. And if you think about it, so it's hard to say whether a species is domesticated or not. I tend to think that it's a continuum between wild and domesticate. And where you are in the continuum actually depends on the strength of the mutualistic interaction between the domesticator and the domesticate. So that's something we are interested in trying to find out. So when can you call a species a domesticated species or domesticate? The best way to do it is to measure the interaction strength ecologically. This is the biologically relevant measure to do. The problem with it is a very difficult thing to measure. How do you measure the strength of it? You can do it with coupled Lotka-Volterra equations, but it's in practice very difficult to do. So we usually rely on markers, either morphological or behavioral markers, to tell us that something is a domesticate or not. This is an indirect way to determine whether something is a domesticated species, but it's easier to measure than interaction strength. And of course, sometimes people use genomic markers to determine whether something is domesticated or not. This is indirect and actually can be problematic because there are a lot of other features of genomic markers that can arise not because of domestication, but because of some other process that we're interested in, okay? This was from a review I wrote several years ago with a former postdoc of mine, Rachel Meyer, who I believe has also visited UNAM in the past. And in thinking about crop species evolution, so looking specifically at plants, domesticated crops, we can think of the evolution of the crop into a series of different stages. The first stage is the onset of domestication. So when domestication occurs, what's really happening? What's happening is that, again, I'm talking about human-associated domestication. Humans are actually intervening in the life cycle of the plant. What they're doing is they're planting it, right? Deliberately planting it and then harvesting the products and planting it for the next generation. That process of cultivation and harvesting actually is what gives rise to domesticated species. And so stage one is this onset of domestication, when humans first began to rely not on hunting and gathering to get their food resources, but to begin to deliberately plant for the next generation. So that's the stage one, the onset of domestication. Stage two is when you actually get, and by the way, archaeologists have called this process the pre-domestication cultivation. This is when humans started the process. The plants haven't been domesticated yet. They haven't evolved yet. They're just being planted by humans. But then you go to this next stage, which is the domestication stage, which is when what you see is that genetically the plant is beginning to change and evolve. There are now several genes or mutations that allow the plant to to live within the human environment much better and to provide these resources to humans. And so those alleles start to increase in frequency. And that increase in frequency is the domestication process itself. And if you were here in my talk in Fronteras last time, you may remember that people had originally thought that this process took place very rapidly, but archaeological evidence and then later on genetic evidence suggests that this takes several thousands of years, especially in the first domesticated species. And then what happens after this domestication process is that you now have a domesticated species that's being cultivated. Now it starts to spread, right? So maize comes from the highlands of Mexico and starts to spread all over the world. And what we'll see later on rice, it starts to spread all over the world. And it starts to evolve local adaptations to the different environments it encounters. That's a process that I call the diversification stage of crop evolution. And then today, stage 4 is when we're doing deliberate breeding as humans. We now, as scientists, do deliberate breeding of crop species. And so right now what we're doing is we're in the final stage of this process of crop evolution. But my lab is interested in stages 1 to 3. That's what we're focused on and trying to understand. And by the way, in stage 1, the changes that are occurring is mostly on the human side. We are changing our behavior so instead of gathering plants, we're actually planting them. We're changing our culture to be able to accommodate agriculture. So that onset of domestication is characterized by changes mostly in the human side and changes in their behavior. And then you see changes on stage 2 in the genetic side. When domestication is actually occurring, now we're beginning to see the evolution of these new crop species that occur, okay? So I don't know if also you remember this. The thing that's interesting to us about crop species from an evolutionary standpoint is that all domesticated species are very recent species. All domesticated species have evolved only in the last 12,000 years, except for the dog. The dog has probably evolved between 25 to 30,000 years ago. But all of the species that we plant in the farm and are harvesting for our food and nutrition all evolved in the last 12,000 years. So in many ways, we are the very early stages of the evolution of these species. And so as an evolutionary biologist, we can try to take the information we learn about how these crops are evolving to learn more about the early stages of the evolutionary process. So my lab works on several domesticated species as evolutionary models. The one on the right, we work on date palms, or dátiles. It was mentioned that my lab has I have another laboratory at the New York University campus in Abu Dhabi in the Middle East, in the United Arab Emirates. And there we study date palms and the domestication and evolution of date palms. But for my lab in New York, our primary focus is the study of rice, Asian rice and African rice, or Rhiza sativa and Rhiza glaberrima. And for the rest of the talk, I'm going to talk to you about our work recently on the evolution of rice. So Rhiza sativa, or Asian rice, you're all probably familiar with it. It's actually it's the major world food crop of the world. It is a crop that is a primary calorie source for almost half of the world's populations. And it's domesticated in China, and the size of the genome is about 400 million bases. So it's a fairly moderate sized genome. And that's nice so that you can do genomic studies with rice as well. So rice evolved, we believe, starting about 9,000 years ago in the Yangtze Valley. Its ancestor is the wild rice, Oryza rufipogon, which grows today in South and Southeast Asia. And from there, humans started to plant wild Oryza rufipogon, and that evolved Oryza sativa, which is domesticated rice. In Africa, they actually domesticated a separate Oryza species, Oryza glaberrima. It was domesticated about 3,000 years ago from Oryza barthii, which is a wild species that grows in Africa. So there's at least 2 independent domestications of rice, an Asian rice and an African rice that has occurred. There's actually archaeological evidence that another Oryza species was domesticated in South America. There's archaeological evidence to show that an Oryza species, there's an archaeological sequence where grain sizes were really increasing, suggesting the process of domestication. but we don't know what that Oryza species is because it was lost. The native peoples of South America stopped growing it just before Columbus came. So we have no knowledge of what this Oryza species is. And Oryza sativa, the species range, this is the areas of the world where it grows, the ones that are colored in the map. So although it originated in East Asia, in China, it has spread through other parts of the world. And in the circle is really what I would consider the original species range of Oryza sativa is within that circle. And I don't know if you know what else is within that circle. So within that circle is where we think rice is grown, or is the original species range of Oryza sativa. And it's also where most of the populations of those areas rely on rice as a staple crop. But does anybody know what else is within that circle? What? Well, yeah, there are bees there, but no, it has to do with the human. Half of the world's population lives within that circle. And it's been suggested by anthropologists that the reason that there was a rapid population growth in East and South Asia was because of its dependence on rice. Rice is actually a fairly high-calorie food crop. But many of you might not know that rice, Oryza sativa actually has a large number of traditional cultivars that are grown all over the world, but primarily in East and Southeast and South Asia. In the International Rice Research Institute, which is the international center for rice research, it's the rice counterpart for CIMMYT in Mexico, which is for maize and wheat. Their seed bank contains seed for 130,000 varieties of rice that have been collected throughout the world. And you can see some of the variation, for example, the seed of Oryza sativa. We use this information and the availability of this genetic collection to work on many of our projects on rice evolution and variation as well. One of the first projects we did was this project looking at, we were trying to understand the relationships between the different traditional rice varieties and wild rice. And this was, when was it? 17 years ago, 18 years ago now. A postdoc in my lab, Ana Caicedo, led this project. And what she did was she did PCR of 110 gene fragments across the rice genome from about 120 traditional rice varieties and about 20 from wild rice, Oryza rufipogon. And the outgroup here was Oryza meridionales and Oryza barthii. And we did a reconstruction of the phylogeny using those 110 genome fragments, gene fragments. And if you can see, what happens is that there's really two genetic groups of rice. There's Japonica rice and aromatic rice groups together in a clade, and Indica and Ausch rice grows together in a clade. And this genetic separation of the Indica rice and Japonica rice had been known for a long time from genetic studies using isozymes and microsatellites and other types of molecular markers. It had long been known that Japonica and Indica rice were separate rice genetically, and actually were suggested to be subspecies of rice. And it had been thought because that they were genetically distinct, these two were domesticated independently from each other or had separate origins from each other. So I showed you that project from 2007 where we had done 110 gene fragments across the rice genome. So we went to the next stage a few years later when what we did is we took DNA sequences across 3 rice chromosomes, chromosomes 8, 10, and 12. And we looked at fragments every 200 kb across these 3 different chromosomes. We wanted to understand how genetic variation changed across a plant chromosome and a rice chromosome. And we did that for both traditional varieties of Oryza sativa and the wild ancestor Oryza rufipogon. And so the data will come out something like this, where we have nucleotide diversity, or pi, and how it changes across chromosomes 8, 10, and 12. And a variety of other parameters that we looked at. So what we did with this data, and we were collaborating then with Carlos Bustamante, and Carlos Bustamante is a human population geneticist. You may be familiar with his work. And one of the things that he developed at around this time we were doing this project, which was the later, about 2009 to 2010, he had developed this new population genomics framework for analyzing population data called diffusion approximations for demographic inference. What he did was he took Kimura's diffusion approximation for the changes in allele frequency of neutral mutations in the genome. And basically what DADI, which was the name of the program that he developed, did was to get the likelihood of a certain data given parameters in this model that you would infer with this data. And so what we did is and you can use this on SNP data. So we took our SNP data from rice, and we decided to test this model here, which is the two origin model, just to show you what this model is. This is Oryza rufipogon. This is the population of Oryza rufipogon. It's evolving over time. And then at one point in time, Japonica evolves from Oryza rufipogon. And then later on, after a certain time interval, indica rice evolves. And then there can be gene flow between different populations. There can be a certain bottleneck period when these species are evolving. So all of those parameters are in the model. And what DADI does is it infers the parameters of the model and calculates the likelihood of the SNP data given that particular group of parameters in the model. We decided to do this on the two origin model, which was what people who work on rice believe was happening in rice. But we also decided to test a one origin model. And actually to this day, I'm not sure why we decided to test a one origin model. It's the simplest model, we just decided to test it. And here you have wild rice, Oryza rufipogon, and then rice is domesticated once as Japonica, and then indica is domesticated from Japonica rice separately. So this is a one origin model. And then when we were doing this, we had this big lab meeting with Carlos Bustamante's group and my group, and he was showing us the results. And the surprising thing he showed us in the results was of those two models, which, by the way, remember what we were thinking of. We were thinking this is the correct model. Okay, this is what everybody believes. He said, actually from DADI, it's the single founder model that is outperforming the double founder model. If you look at the likelihoods of the single founder model, it's a higher likelihood than the double founder model. And I didn't believe him because I said, this is impossible. Everybody knows rice evolved twice. You cannot have the single founder model work. I said, maybe what's the problem is that you're not incorporating selection in your model. And so why don't you incorporate selection and see what happens? He says, okay, they did that. They incorporated selection, and it's still the likelihood for the single founder model still outperformed the double founder model. And I didn't know what to do with this because I said, nobody's going to believe that. By the way, if you know Carlos Bustamante, he's a MacArthur. He won the MacArthur Genius Prize. So he's a genius, but I still didn't believe him. Because everybody knew that Japonica and Indica evolved separately. And so how could you say there's a single founder model? So Jean Molina, who was the postdoc in my lab, who was working on this project, then came to me and said, Michael, what I want to do is I want to reanalyze all of the previous data that showed that rice had originated twice, because something must be wrong here. And I said, no, I think those studies are right. I think Carlos is wrong. But he did do that. And she found that there was a flaw in all previous studies that included the studies that we had done. So many of the people here probably know systematics and know, when you're trying to look at doing the phylogeny of a group of very closely related populations or species, what do you do, right? You don't rely on one marker. You don't rely on two markers. You try to rely on as many molecular gene sequences as you can, 10, 100 markers that you do it. But what do we normally do, at least in the old times, we have DNA sequence data from, let's say, 20 markers. And we know we're looking at very closely related species. We concatenate the markers, produce them as one data file, and then run them as if they were one gene, right? At about the time we were doing this study, or when we had presented this study, there was a paper in Systematics Biology that showed that if you do that for closely related species, there's a 75% chance, in some cases, 75% chance, you'll get the wrong answer, okay? And so at the same time that this was happening, a new method came out from Oxford, the BEAST method, the Bayesian evolutionary analysis of sampling trees. And as you're familiar with this method, what it does is if you have 100 markers, it doesn't concatenate the markers and believe that it's just one gene. It makes the trees from each marker separately and then integrates after looking at what each marker is doing. And what Jean did was she took all of the previous studies that had shown two origins of rice, and these are the studies, the number of markers used, total number of sites, blah, blah, blah. And she ran beast on them. And when she ran beast on them, except for one case that was ambiguous, in all cases, the support was for single origin models. So all of us had done it wrong before. And when you use beast, you were getting a single origin model, right? So based on this and also based on archaeological evidence, what we suggested was, what happened was 9,000 years ago, Japonica rice evolved in the Yangtze Valley. That's based on archaeological evidence. The oldest examples of rice is the Yangtze Valley in China. In India, they were either, at about the same time, they were either harvesting wild rice, they were doing something, or they may have started to plant rice, but it hadn't been domesticated yet. And then Japonica evolves, or Rhiza sativa, it becomes domesticated. It spreads towards East and Southeast Asia. And then Japonica makes itself to India about 4,000 years ago. And what it does is it hybridizes with whatever they were doing in India. And that gives rise to indica, okay? And if you look at archaeological sites, you'll know that this made sense, because if you look at China, for example, where you can see clear archaeological evidence of cultivation and domestication of rice, they're very old. They're between 6,000 to 9,000 years ago. But in India, actually, you don't start to see rice until fairly late, until about 4,000 years ago. There's some very sparse examples around the archaeological record, but it's only after 4,000 years ago that you start to see an increase in rice in the archaeological record. And by the way, it starts to increase in India archaeologically at about the same time that you see artifacts in India that are clearly influenced by China. And so so the feeling was that rice came in with other artifacts from China into India starting about 4,000 years ago. Our suggestion was it hybridized, and that gave rise to China. So that made a lot of news when we published this in, I forget when we published this, in 2010 or 11. It's very funny because, as you probably know, there's a lot of national fervor that goes with who was the first one to domesticate what species. You're lucky that in Mexico, nobody claims. Corn was definitely domesticated in Mexico. Rice, there was always a problem, but they would say U.S. study sparks row between China and India. The U.S. study backs China, Great Indian Debate. Oh, by the way, in the paper we published, there were both Chinese and Indian researchers, so we were covered by that. But that model that we presented was still a problem because a lot of people didn't believe it. And in fact, there was a letter to the editor in the Proceedings of the National Academy saying the Peruginan lab is wrong. And then a year later, a group from the Beijing Genome Institute published a separate study with a separate, completely different approach, and they came to the same conclusion we did. What they did was they looked at domestication, loci of domestication genes, and in fact came with the same model we did, that it was domestication of Japonica, it crosses something that gave rise to indica rice. So there was a separate study that the Beijing Genome Institute did. And so together, and now this is kind of the accepted model. We think it's the accepted model for how rice was domesticated and evolved. Japonica came first. It integrates with something in India that gave rise to indica rice. And by the way, the model that is supported by the fact that if you look at the genes associated with domestication in Japonica rice, in Rhiza sativa, and we know what the genes are. Some of them are the shattering genes, prostate growth, and LABA1. If you do the phylogeny of those genes, the alleles in indica rice actually groups with Japonica rice, suggesting that it was not a separate origin, but it came from Japonica itself. So this was a study. This was from the Chinese study, but it was confirmed by a postdoc in my lab recently, Jay Choi, about 7 or 8 years ago. And he also did a model which showed that there was definite evidence of gene flow from Japonica to indica rice that was occurring. So we now know that hybridization and introgression was an important component of the evolution of indica rice. And so indica rice, we think, is really a hybrid between Japonica rice and something that they were doing in India. Okay, so that hopefully gives you a little bit of a story of how we think rice originated and was domesticated. For the last 8 years, 7 years, my lab has been really focused on trying to understand how rice spread after it was domesticated. And if you think about where it spreads from temperate to tropical regions and across a large gradient. And trying to understand the evolutionary spread of crops, I think, is the next major step in trying to understand crop evolution. And in order to understand this, we need to understand things like how did humans migrate themselves, because they were the ones that were bringing the plants with them. How did different cultures interact? How do plants adapt to different climates and day length and different environments and biotic interactions? So there's a lot of different questions that you can try to answer in trying to understand how crops themselves have spread from their centers of origin to other places. And by the way, the other thing about this is really a question of adaptation. And so in this time of climate change, we might be able to learn how crops can adapt to different environments and adapt to different climate zones, for example. So that's kind of the long-term idea of this program. But first, we have to figure out how rice, for example, itself has spread. So about 6 years ago, we started a project where we took genomic data from rice to try to infer its population structure and its pattern of dispersal across the world. We started off with the genomic data that's available from rice. This was, again, from the Beijing Institute of Genomics. They've sequenced 3,000 varieties of rice. So we have data from 3,000 varieties, of which about 1,300 or 1,265 are traditional varieties. By the way, this is being superseded. We're actually involved in a consortium. We now have data for 20,000 varieties of rice. That's going to be published. It's going to be submitted in about 3 months. So this data set is getting bigger, but this was the original data set that was important. And we had 1,265 land races or traditional varieties from this project. And then we also sequenced 178 land races in our lab itself. So we contributed data from ourselves. And so if you look at where these varieties are from, these are the dots are where we get these traditional varieties from in East and Southeast and South Asia. And then when we did a principal component analysis of the SNP variation, you see a PCA that looks like this, where there are 3 major groups of rice that you can see. This is indica rice on this group here. This is japonica rice. This is actually Aush, which is another subspecies that is localized primarily in Bangladesh, but it's grouped as a separate group. This is aromatic rice, like basmati rice or jasmine rice we can get from Thailand. These are the aromatic rice. It turns out the aromatic rices are a hybrid between Aush and japonica rice. So that's another example of hybridization, giving you another major subspecies or variety group. So we focused our efforts initially on japonica rice because, as I said, we thought this was the original domesticate. So if you look at that, there are 372 japonica land races in that corner over there. And if you expand it, this is hard to see, but it expands. This is only japonica rice, you can expand it into different groups. And actually, if you know what you're looking for, it makes sense. This is temperate Japan. This is from China, from Japan and Korea and Northeast China. These are japonica rice that grows in temperate regions. This is from mainland Southeast Asia, tropical mainland Southeast Asia. And this is from island Southeast Asia, from Indonesia and the Philippines. And then there's another group here that groups with, it's a Japanese group that grows in the uplands. So it's still unclear what that group is. But this gives you the populations. You have a temperate Northeast Asian group, one of which grows in the uplands. You have a Southeast Asian mainland group, and then Southeast Asia, Indonesia, Southeast Asia, Philippines. So the interesting thing about this is that it's a crop, and you think that crops move very quickly or very easily across countries. They're grouping by geography, suggesting that there's a geographic signature to the evolution of these crops. One of the things that, this was a postdoc of mine who led this project, Rafael Gutaker, who's now at the Kew Botanical Gardens. And he applied a method that's used in human population genetics, which is admixture mapping. And what, not admixture mapping, but admixture population genetics. What it does is it tries to look at the relationships between populations, but it includes gene flow in the model so that it's not just bifurcate trees, you can include gene flow. And this was the model that he came up with, which is very complicated, I understand, but you can see, population splits here, for example, between this Indonesian population and these Philippine populations. And you see with these dashed lines are gene flow events that are contributing to the movement of alleles between geographic populations. It's a very complicated diagram. But what we did is we took that information and the geography, we knew where the land races came from. And we also took archaeological information over time. So this shows you where rice grains are found in the archaeological record, starting about 9,000 years ago and going later and later in time. So 9,000 years ago, there are only a few of them. Then you're seeing more of them between 8,000 and 7,000 years ago, and so on and so forth, until about the last 1,000 years, they're all over the place from the archaeological record. So we were able to incorporate archaeology into our data. And by the way, the reason for that is archaeology is the paleontology of crop science. It gives us our fossils for our work. And so with all of that data, we came up with a dispersal model for how we think rice moved in after it was domesticated, Japonica rice. It's complicated, I know, so I'm going to do this simply by looking at things one by one. So I'll lead you through the story. So we think domestication occurred 9,000 years ago in this region, the Yangtze Valley, the middle Yangtze Valley, China. That's where the oldest archaeological evidence comes from. That comes straight from archaeology, that's come from genetics. The first split we see is a split of the tropical and temperate Japonica. Temperate Japonica, so if you go to a Japanese restaurant and you have sushi, you're eating temperate Japonica. That was the first major evolutionary split between tropical and temperate. The tropical Japonicas are the ones that went south into South China, Southeast Asia. And if you look actually at, again, at the phylogeny that we did 18 years ago, temperate and tropical Japonica are actually phylogenetically distinct from each other as well, which reflects that split we think that occurred. So that's the earliest split that has occurred. And by the way, now we can, and this just came out, literally this came out this morning. We're redoing some of the analysis, and my graduate student, Ornab Alam, was doing some of this. He's defending his PhD in 3 weeks, so he's still finishing up some of his work. But he's able to date when those split times occur in that phylogeny. And what we find is this split time between temperate and tropical Japonica occurs between 4,300 and 5,450 years ago. That's the uncertainty bars around the split times. And that's interesting because if you look at the climate of East Asia during the time, this is a period of time when temperatures were actually going down in Asia. So this is a graph from an archaeology colleague of mine, Jade Guedes. This is in calendar year, so this is 8,000 BC. Where you see the shaded is where rice is found in the very northeast part of China, the Shandong Peninsula, the one that's close to Korea and Japan. And you see that early on, about 8,000 to 7,000 years ago, you see rice in the Shandong Peninsula, and then it disappears. And the disappearance is coincident with the fact that temperatures are starting to drop in that region. But before that, essentially what's a tropical crop is growing in northeast China. And then it reappears starting about 4,500 years ago. It reappears back in that area. So there's a period of time where it's not found there. This is a period of time where you're seeing a decrease in the temperature of that region. And this is the time where we think that the split of temperate and tropical Japonica appeared, okay, based on our genetic data. So there seems to be a coincidence between the falling temperatures in the region, the sudden development or evolution of temperate Japonica, and the reappearance of it in this region, which is now getting colder and colder, until, of course, we got the global warming at the very end, at the bottom there. Unfortunately, I don't think this is, yeah. This was supposed to be an animation. Oops. It went out. We need to. Oh, no, I got it. It came back. So I think it's fine. Hopefully it'll be fine. Okay, sorry. That was supposed to be an animation, but unfortunately it's not working. What it is, it shows you, I'll show you this graph at the bottom. This was done again by Jade Guedes. What she did was she took tropical japonica, and what she knew about the number of growing degree days you need for tropical japonica to flourish. And she showed that whenever you see this yellow, it could grow in that region, given the known temperature. This was 5,500 years ago. So 5,500 years ago, it could grow pretty much all over Asia, except for the Tibetan Plateau. So rice could grow. But as you go forward in time, the temperature starts to drop. And so the probability that you're going to get tropical japonica to grow in this region starts to drop quite a bit. And that was what that graph was showing. And as I said, you can see that kind of in the archaeology. About 5,000 to 6,000 years ago, rice was grown only in this region up north, in the same latitude as Japan and Korea. But starting about 5,000 years ago, you're starting to see rice move down to the tropics. And that's a period of time where I think temperatures were going down. And so rice had two evolutionary responses. One was to evolve to temperate japonica. So temperate japonica was able to stay in the northern regions, but then rice had to move, the tropical japonica had to move southwards in the more tropical areas. And that's when you begin to see movement of rice southwards and that. So that was the first split, the temperate tropical japonica split about 4,000 to 5,000 years ago. And then mainland and island Southeast Asia, the diversification of that occurred very soon after that. As rice began to move down to the tropics, there were populations established both in the mainland of Southeast Asia and the island part of Southeast Asia. And within island Southeast Asia, within the last 3,000 years, you start to see the movement actually of rice and the diversification of rice in the different islands of Southeast Asia. So we're able now to put together a picture of how we think rice has evolved in East and Southeast Asia. I'll tell you one little twist to this story that some of you may, especially if you know world human migrations well. One of the biggest and the last human migrations was the Polynesian migration into the Pacific Ocean. And the major driver of that was a group of people from Taiwan, the Austronesian people who were in Taiwan, who went out of Taiwan, went down to the Philippines and into Polynesia as one of the last major human migrations in the world. What some of you may not know is that Peter Bellwood, is a famous anthropologist in Australia, has suggested that rice and rice agriculture came to Southeast Asia as a result of the Austronesian expansion. So when the Taiwanese Austronesians were moving to the Philippines and into Southeast Asia and then into Polynesia, they brought rice and rice agriculture with them. And he based this on linguistic grounds. All of the Austronesian languages in the region have the same word for rice. And that's why, and rice agriculture. And that's why he suggested this. We decided to test this in a strange way. I gave the presentation I just gave you, I gave in Taiwan several years ago. And after my talk, Caroline Xing, who was a member of Academia Sinica in China, came to me and he said, Michael, and we had collaborated in the past, so she knew me. She said, Michael, I actually have DNA sequence data for japonica varieties that are grown by the Austronesian peoples of Taiwan. So if you don't know the island of Taiwan, you think of it as part of China, or that's not part of China, but it's colonized by Han Chinese. That colonization is actually fairly recent. The original indigenous peoples of Taiwan were Austronesians, and then the Han Chinese came in much more recently. And then, of course, after Mao took over in the mainland China, that's when you had a big influx of Han Chinese. But the original inhabitants were Austronesians. And she had gotten the varieties that the Austronesians in Taiwan were planting and sequenced them. And at the same time, I knew that I actually had varieties that had been that we had sequenced from Japonica land races in the northern Cordilleras of the Philippines, where actually another indigenous group that were Austronesians, the Ifugao groups, were planting rice in these big rice terraces. So I said, well, you have your data from Taiwan, I have my data from the Philippines, let's put it together. And let's test Bellwood's hypothesis. And when you did that in this phylogeny that I showed you earlier, you show that the Taiwan indigenous land races come with two populations, Taiwan 1 and Taiwan 2. And so in Bellwood's hypothesis, Southeast Asian and the Philippine varieties must come from Taiwan 1 or Taiwan 2. By the way, those northern Philippines varieties, they're shown here in this part here. And you can already tell they're fairly distinct. And why is that? When we did the admixture graph, and just to orient you, this is the temperate tropical split. This is the mainland island Southeast Asia split and island Southeast Asia diversification. This is Taiwan 1 and Taiwan 2. They're both related to temperate Northeast Asian land races, the ones you use to make sushi. And they have gene flow from either the mainland Southeast Asia or from the northern Philippines. So it's actually the reverse. It's not that Taiwan is providing rice to Southeast Asia like the northern Philippines. The northern Philippines is providing alleles to the Taiwanese land races. It's going up south. And so what we believe is happening is Peter Bellwood and his idea that rice and rice agriculture comes into Southeast Asia from Taiwan is wrong. We think that Japonica rice came from mainland Southeast Asia into Indonesia and then starts to go up north. Temperate Japonica gets to Taiwan, and then some tropical Japonica from the Philippines gets to Taiwan. They hybridize, they form the indigenous land races that the Taiwanese are doing. So they're a recipient, they're not a source. Okay, so that kind of gives you the story of Japonica rice, which is the oldest rice that we know of. Just very briefly, we also did indica rice. We did the same thing. We did indica rice, which is from India, and we looked at the geographic structuring, and we can find nice geographic structuring. Actually, I'll return to this one. The problem with this model, and we wrote about this in our paper 5 years ago in Nature Plants, is that this model is not stable. So every time we run the model, we get a different group of relationships. We can't stabilize it. The only thing we can stabilize is the split of Indian indica and China Southeast Asian indica. That's the only one that keeps on being stable. So in our paper, we didn't talk much about this because we didn't have a stable model that we could work in. Much more recently, a graduate student in my lab has added more data to the indica story. And the other thing he's done a different method. He's using a Bayesian method to look at the relationships in the populations. And he's finally gotten a stable model. And in a stable model, you have India, indica rice, and then China, Southeast Asia rice. So China splits from Southeast Asia, mainland Southeast Asia, and then Indonesia indica comes much later. So it makes sense geographically. You can't think of a stepping stone population model for the evolution of indica rice. He's also able to date the splits. They're very recent. They're only in the last 2,000 years. So the movement of indica rice out of India into Southeast Asia occurs very recently, only in the last 2,000 years. And we think that's because Japonica rice was already being grown there. And the people in the region didn't see the need to get indica rice. Although I must say now most rice in South and Southeast Asia is indica rice because it's actually very high yielding compared to Japonica rice. We were able to date it. By the way, archaeologically, we know that indica doesn't make it to central Thailand until 1,700 years ago. So our archaeology, and by the way, one of the stories here is that when you do population genomics well, the genomic data and the archaeological data line up really nicely together. So that's really good to know. Okay, so I'll give you this last few slides. All of the stuff most of the stuff I've shown you has been published. This one is not, and again, this was being finished by my student Arnab Alam, but I think it's of interest, so I'll tell you what it is. The nice thing about working with rice is that there's a lot of genetic information on rice, and there's been a lot of genome-wide association studies. So we know a lot of the mutations that are involved in variation in rice and diversity. What gives you the grain color, the height, the flowering time, and a lot of things. There are actually 200 plus functional mutations identified in rice. This is functional mutations that are segregating in traditional rice populations, right? 73 of these are segregating in Japonica at greater than 10% frequency. So they're fairly moderate frequency. And so we know what the genes are and what the mutations are. And the nice thing is that recently there's been a new development in population genomics methodology, which allows you to do several things with known mutations. They allow you to date, for example, let's say this mutation here in this coalescent 3. If you have this mutation, these relate is a method that allows you to estimate the age of that. How old is that mutation? And the other thing it can allow you to do is reconstruct the change in frequency of the allele over time in the past, even in the absence of ancient DNA. Okay, that's something you can normally do with ancient DNA, but even in the absence of ancient DNA, you can do this. And so Ornov, my student, decided to use this on those 73 functional mutations in Japonica. And this is something that we found interesting. What you see here is time. This is a log plot. This is 0. This is the present. This is 1,000,000 years ago. Each of these is a separate functional mutation. Some of them we've put names to. This is a known mutation in rice that is important in phenotypic diversity. These gives you the uncertainty around the origin. This bar here is the separation of the Pleistocene, or the last ice age, and the Holocene. And Oryzis sativa and all domesticated crops evolved in the Holocene period. Almost all of the majority of functional mutations that in rice that are important in phenotypic diversity in current rice land races and are being used by breeders to improve rice originated in the last ice age. They're old. They didn't originate during the time of the origin of rice. There are very few mutations that originated during the Holocene. By the way, the ones here on top are ones where we don't know. We're uncertain whether they originate in the Holocene or the Pleistocene. But these are all the Pleistocene mutations. These are the Holocene mutations. You can tell there are more Pleistocene mutations than Holocene mutations. The mutations we're using for breeding, the mutations that were important for domestication and diversification are old mutations. They're not new de novo mutations. The other thing that was interesting, he took one mutation that was involved in non-shattering. So some of you may know one of the key indicators of domestication in cereal crops is non-shattering. That is, the seed stays in the plant when you harvest it. Most plants are shattering, that is, when you harvest, when the seed is ripe, they fall off. In domesticated species, you evolve non-shattering because when you harvest the plant, you want the seed to remain on the plant so you can harvest them. So one gene that's very important in non-shattering, in the evolution of non-shattering rice, is the QSH3 gene, this mutation in the QSH3 gene. This is a mutation that's known. Using the method that Ornov did, this is the allele trajectory of QSH3. It starts to rise about 8,000 years ago. Remember I said Japonica started to be domesticated 9,000 years ago? Well, he sees that this mutation is starting to increase in frequency 8,000 years ago. What are these dots here? These are the frequency of non-shattering seed that is found in the archaeological record. So early on, 8,000 years ago, you're only finding 20% of the rice seed in the archaeological record is non-shattering. About 4,000 years ago, they're about 80% or near fixation. And you can see what's happening here. The non-shattering allele is increasing in frequency. It's aligning really nicely with the archaeological record on the frequency of non-shattering alleles. So it's a nice superposition. So we can use this to maybe identify other domestication alleles, right? So if we know that QSH3 is evolving as a domestication allele, what are the other mutations that are rising at about the same time? And we're able to identify a flowering time gene and 2 plant architect morphology genes, Teosinte branch 2 and SPY that are involved in plant architecture are also rising in frequency at about the same time that QSH3 is rising in frequency. So suggests these might be domestication genes. And you can also say that some genes that were thought to be domestication genes are not domestication genes. GS3 is a mutation that's involved in grain size, in increasing grain size, so it's thought to be a domestication gene. It actually starts to rise in frequency much later than we know. So it's probably not a domestication gene. It's probably something that came in later. So with these new population genomic technologies, we're now able to date and to try to correspond the rise of a frequency of a gene with the rise of a certain phenotype or a certain event. Finally, remember the rise of temperate Japonica, the appearance of rice in Shandong at about this time. These are genes that, most of these genes are associated with cold tolerance, temperature tolerance in rice. And this is the time when you can see the appearance of Shandong. The rise of the genes associated with temperature tolerance or cold tolerance in rice starts to rise up after rice starts to reappear in the northern areas of China. So that again is a nice correlation. There's one flowering time gene. That, by the way, I showed you that most functional mutations in rice originated in the last ice age in the Pleistocene. The exception are the flowering time mutations that change the flowering time genes. Those are younger mutations. Those are Holocene mutations, and they also rise up in frequency very late in rice evolution. So in this seminar, I was able to tell you several things. I know it's a lot of information, but I hope you take with you these types of these ideas that rice was originally domesticated in the Yangtze Valley, Japonica rice, and indica arose via hybridization of Japonica to something that was going on in Southeast Asia. That this global cooling that started around 6,000 years ago and continued on for a long period of time is associated with the dispersal and diversification of Japonica rice. Indica rice is dispersed much later than Japonica rice. And that this one, the last few slides I showed you was very new, literally in the last week. Most functional mutations in Japonica rice are Pleistocene in origin. They're not new. And we can correlate their increase in frequency with various evolutionary and climate events. And so I'd like to thank the people here that are shown in asterisk are the ones who have contributed to the data that I gave in the talks and some of the collaborators in the project. And I'll give a shout out because she's a UNAM graduate, Sauro Rosas, who's from the Institute. And she's a postdoc in my lab now who runs the ecosystem adaptation project in my lab. So thank you very much. And I'm happy to answer any questions. Any question?
Hi, thank you for your talk. I'm always happy to see the most parsimonious hypothesis being correct, right? Or at least the most supported one with the single origin. I have a couple of questions. The first one is your definition of definition of domestication. There's a keyword there that is coevolution, right?
Yeah.
And you kind of show us the evolution or the change in one of the species involved in this interaction. So yeah, I would be curious to know if someone has a study, the other half of the interaction.
On the human side, there are two answers to that question. One, on the biological evolution side, so people have been looking at genes associated with diet changes, right? With the change in amylase copy number, salivary amylase copy number, and of course lactase, adult lactase tolerance. So more effort is being done to look at diet changes associated with that. So that's number one. Number two, which I'm in the process of thinking about, is the other major changes, of course, at least for us, for humans, are the cultural changes. So they're still evolutionary changes, but they're cultural, they're not biological. And to what extent are the changes we're experiencing, the evolutionary changes, how much of that is cultural versus biological? It's a big question. So
okay, interesting. Now, this problem that you're trying to solve is a very recent, in terms of evolution,
right.
Evolutionary time. And you mentioned how concatenation failed.
Yeah.
I was looking at my colleague here because we teach systematics together.
Yeah.
And you mentioned that concatenation failed at this hypothesis of the one origin or the two origins. And I will say in defense of concatenation that I think choosing your outgroup in this kind of questions is very important. And that's one of my questions, like how do you root trees in this study of rice? What do you use?
Well, we did an outgroup rooting from using two species that were, well, they're definitely clearly not in the range of sativa and within sativa and rubipogon. The paper in systematic biology that suggests about concatenation, there are only very specific instances where it fails. So it's not like it's a general failure. And I still think that you can concatenate if you meet some of the criteria they had. But there are certain, and I have to say, I've forgotten what the certain criteria where they can fail quite dramatically.
Mhm.
It so happens that ours was one of those that failed.
Yeah, and I mean that is split between the two sister
Yeah.
species is so recent
Yeah.
that it could be
Maybe-
difficult to find synapomorphisms.
It would be interesting to rerun this with an even more distant outgrowth to show, because we're still relatively close to Sataiba here.
I think my last question is, like, you've seen, how difficult this question is. You talk about this software called Relate that allows you to date mutations. Aren't you skeptical of those results?
Well, so we're always skeptical of those results. That's why we try to correlate it with archaeology, because we think archaeology is our ground truth. Archaeology has its problems, but it provides another independent level of evidence. And so I get nervous when when archaeology and genomics doesn't meet up. But so far it's been they've been good at meeting up. So I'm less skeptical.
Thank you, Michael, for your wonderful talk and for coming to Mexico. Bienvenido anytime. I have a question about, so I'm thinking all the time about maize. So during domestication of maize, it's not one single crop that was domesticated, but a bunch of crops that, they are grown, traditionally together. So I was wondering if there is a similar pattern in rice that, there are several crops that are grown together. But so when you see migration, you don't see only migration of one single crop, but a whole bunch of species migrating.
I haven't thought of that. I mean, usually you see a cereal grain and a legume grown together. The legume that would be most grown, at least for Chinese, would be soybean. I haven't thought about that as a package, actually. I've been wanting to do an analysis where we do comparative population genetics of different crops in the same region in China, but we haven't done that. It would be soybean, but I think soybean is a little later than rice, but I'm not sure. But I think what would be interesting, as I said, is the next area of study is to not look at just one crop, but multiple crops simultaneously, and see how they're evolving together or not.
Yeah, that's what we see in the corn is the milpa system. So it's a bunch of species.
It's right. It's squash, it's corn, and it's bean. And the beans and cereal and legume package is known throughout, that most cultures will put together a cereal crop and a legume. I just don't know what it is in North, because in the Fertile Crescent, it's wheat, barley, and lentils is what they put together.
And a second question about the technology. So when you show the places where rice is grown, I often see the terraces that are flooded, and so I guess that requires quite a bit of technology, to build them. And so is there any correlation with that migration of the technology or archaeological evidence that allows for people to adopt rice or migrate it?
Yeah. Again, that's a problem in these types of studies. What we lack is the ethnography associated with this, and we don't have the data on the ethnography. So we have good genetic data. We don't have the associated ethnography map data, and that's what you need. But that's going to be really hard to do. So right now we're just tracing rice grains as they're moving in the archaeological record. So, okay.
Just a question about the African rice.
Yeah.
Do they still grow them?
Yes, they still grow African rice. African Rhiza glaberrima actually is much more resistant to abiotic and biotic stresses, and they still grow them.
So, long time ago, I read that African people that were brought to the Americas, they brought their own rice, and this was the African. And how much of could it be some of those rices that you're dealing with hybridization with African rices.
Right. So we did a study on African rice in the Americas. So they're still growing them in Suriname.
Yeah.
There's a whole really fascinating story about African rice in that. We're starting, so Ornov, the student who did the Japonica rice project, is in the next phase of his project is to look at the movement of rice outside of Asia. So he's looking at Europe, Africa, and the Americas, and see what's going on there to see if there might be evidence of hybridization. The African rice story is interesting for a number of reasons, because the in the United States, the economy of the Carolinas was based on rice agriculture, and there's very good evidence that they purposely imported slaves from West Africa that knew how to grow rice based on their experience with African rice, and that developed the Carolina plantation economy. But while they were growing Asian rice in the Americas, the African slaves brought with them some of the African varieties. And as I said, in Suriname, in the interior part of Suriname, they're not indigenous peoples. They're a group of escaped slaves called the Maroons, and they are planting African rice in their fields.
Yes, that's what they do with what they call beans here in Mexico.
Oh.
So the ones that they grow, they are dignas and not faciolos.
Oh, okay.
So, but I was wondering about this migration of groups, which is this one was really huge because we're more than 1 million Africans coming to the Americas,
Right.
and they brought a lot of different things with them.
Yeah, and as I said, they did bring African rice with them.
Yeah, and beans too
But
from Africa.
they only use it very limited way. They use it a lot in rituals. They still have, not for widespread consumption.
No,
Yeah.
not at all.
No.
Okay,
Yeah.
thank you.
Thanks for your talk. I was wondering, now that you're doing this big survey regarding land races, you're including a lot of land races from different rice varieties, I guess. How much are you finding those? I know it's not maybe the focus of what you talked about here, but you're probably finding local hybridization with other Oryza species. I don't know if that's common for rice, or you haven't looked into it.
We haven't looked at that. And the place to look at that is in Latin America, because you have Oryza glumapatula here, which can hybridize with Ivan. It's not clear yet. Rod Wing, who's in charge of the 20,000 Rice Project, is very interested in looking at if there's any evidence that glumapatula has gone into sativa, or the other way around. That is not clear. Yeah.
And the next question is, how much, like in Mexico, there was this huge discussion about the unicentric or polycentric origin of maize, long ago, and that was settled some 30 years ago.
Yeah.
But more recent studies, and the whole discussion was based on cytogenetic data, where they saw that some lineages of land races in Mexico, their chromosomes were more similar to different Teosinte species, So that was one of the bases of why some people proposed that maize was domesticated several times.
Yeah.
And then that was, in a sense, put to rest. But what they've found in later works is that the single origin still keeps, but it seems that once it started diversifying, it was taken to other parts of Mesoamerica and South and North America. These original, let's say, domesticate variety hybridize it with local Teosintles. And that, I think, seems to explain why cytogenetically you can actually see that the chromosomes are different.
I'm beginning to hear those types of studies for maize, and that would be interesting, because one of the things that we have to get away from is this idea that domestication is an event that just happened this one point in time. It's a process that's continuing. And among archaeologists especially, they think of it as a landscape process. This is something that's occurring over a large landscape, and maybe it's much larger. It includes where it moves outside of Mesoamerica. So it's a much more dynamic process than just a single event, and that it's occurring over a period of time. And the period of time during domestication, as I said, it's not decades or hundreds of years. Rice took 4,000 years to be fixed. So I don't know what the number is for, or what the idea is for maize, but it probably looks several thousand years, and there's a lot of possibilities that could happen there. So yeah, so it would be interesting. Actually, that would be interesting if that becomes a recurring theme. Maybe we've stumbled into another pattern here in domestication.
And then just one last very general question, but you're a population geneticist.
Yeah.
How easy, well, okay, not easy, but I understand it's easier to make population genetics inferences when you're working with annual crops, just given that, they evolve, their generation times are shorter. So you have more generations, you can look into it. How well is the evolutionary history of perennial crops being put together?
That's on our date palm project, we're still struggling with that for two reasons. Like one is we don't know the generation time. Like we can infer the generation time, but it's not clear what the generation time is, which makes estimating dates hard. And then if the generation time is 5 or 10 years, right, which is possible for perennial crops, there's not been a lot of time since the domestication to the present, probably 200 generations. So we're still, so I still have a former postdoc working on the date palm population genetics. She still hasn't given me answers yet on this. So I don't know what to expect for perennials.
Thanks.
Yeah.
Just one last question before we have coffee.
Thanks for your talk, Mike. What prevented rice migrating west? I wonder, it was 4,000 years in the north.
Right. And why didn't it go west?
It spread south, but not west.
Well, there are two questions. Well, there are three answers there. Well, one is it did go west by the old Silk Road. When I said that Japonica rice gets into India, it doesn't get through India through the east. It actually gets through the northwest of India, and that's where it starts. And you see the first Chinese influences in South Asia in the northwest, not in the east. And my colleagues have done an inference on possible routes of rice into India from Japonica rice, and it's the northwest. So they do go to the west. The problem is a lot of Western China is desert, so they're not planting rice. They can bring it with them, but they don't plant. It does get to the Middle East by about 2,000, we think about 2,000 years ago, 2005. The Romans were eating rice that were imported from them, and the rice was grown in Iran and Iraq about 2,000, 2,500 years ago. So it does go there, but it's not as much as going south.
Yeah, but it took too long to go really west. I'm thinking on this. In maize was the opposite. It went from the tropics to temperate regions.
Right.
And it stayed in the tropical America for at least 4,500 years.
Yeah.
Before crossing the Tropic of Cancer in Mexico. It takes 2 months by foot going from here to Zacatecas. Yeah. It took 5,000 years to appear in the archaeological record, maize. It took only 1,000 years to go all the way to Peru.
Right.
After the first initial time of domestication. So photoperiod seems to be a really strong
photoperiod
selection
is very strong, but if it made it all the way to Peru faster than it made it north, I mean, that's still a big latitudinal change.
But photoperiod is pretty similar. It's tropical.
So we know that that's why the flowering time genes are important in dispersal, because they control photoperiod response. But the other thing that, it might just be cultures just don't like it, because at least for rice, right? The culture in China for humans in terms of culinary is they boil rice, right? That's what you do. The Middle East, they're a baking culture. They really don't work. So it may also be that it takes time for the cultures to adapt. That would be an interesting question to ask, but this is an interesting question. Why did it take so long to go up?
Yeah, and it took about also 4,000 plus more years
Wheat and
to go
barley.
south for rice.
Wheat and barley, when it got out of the Fertile Crescent, it stopped in the Balkans for 2,000 years, and it had to mutate the photoperiod genes before it could go into Northern Europe. So at 2,000 years, it stopped in the Balkans. It couldn't move because of the photoperiod response and vernalization response genes. So, yeah, that's interesting. That's why crops are interesting to study. We have these questions.
Yeah, there must be lots of photoperiod genes required to really break the flowering time.
The HG genes that I was showing, the flowering time genes, they were all photoperiod sensitive genes. So, they were involved in photoperiod sensitivity.
Thanks.
Let's thank our speaker, and you can have more questions. Next topic.